Are you gonna open up the opportunity to submit the next study session soon?
Say again?
Are you gonna open up the opportunity to submit the next study session for this week?
It should be open for this week. Is it not?
I don't know. I usually get my assignments like straight from the grade book.
Let me check in a minute, okay? All right, I think I'm ready. Let's go. And close that down. Oh, it isn't open because I don't have it published. Let me publish that. And it's not the right date either. Come on, where's the date? That means I probably have to do that for all of them. I just opened it now. Well, click that save. Now I opened it and I see what the problem is. There was a problem last term where the grade book wasn't adding correctly. And I was going to move the extra credit study session totally out of the class. And then instead of having it, I don't know, 9 or 10 of them, I was going to just do one. And you guys were just going to do it all over using that one. And that would be easier for the computer, help people to figure out what was going on. Because there'd be less thing in my my class. Anyways, so all the others are shut down, it looks like, except for the first one. So I'll need to turn on the others. If they don't get turned on, please somebody remind me. Because I've got to change the dates of them too. The dates aren't right. All right, any other questions? If not, let's go ahead and talk about the lab. Well, no, I got to turn on the browser first. Screen recording. Share the screen. We'll optimize for videos. I'm not sure I'm going to use a video, but oh, good. The lecture finalized. What am I looking for? There we go. All right, today we're doing lab 4. Note this week we're only doing one lab. So that means on Thursday, I will show up for questions and answer. But as soon as the last student answer is answered, I'll log off. I will be there from 6:30 to 6:45, regardless if anyone shows up to answer a question. But on Thursday, there is no lab. Any questions about that? What you can work on instead of working on a lab is the infectious disease project. Later in the term, after we start the unknown project, you can on Thursday work on your unknown project. All right, any questions about any of that? I might have it here. I did put a discussion part, so if you want to talk about the lab, there's discussion. It's right there. I'll come back to that. All right, that's it for what we're doing today, lab 4. So let's talk about lab 4, staining techniques. There are 3 different stains: a simple stain, a differential stain, and special stains. There's some reading you should do in the textbook. I'm not going to discuss it. It's actually quite a bit to discuss, but actually most of them are very little reading. It's just that there's a number of them, but it's all in different types of stains. There's some video clips and there's a virtual lab for today. Upon successful completion of this lab, students should be able to prepare a bacterial smear for staining, understand the principles and procedures used when performing a simple stain, a differential stain, and a special stain. Be able to interpret the results for a simple, a differential, and a special stain. And then lastly, be able to define the terms: a simple stain, a differential stain, a special stain, what's a coccus, a bacillus, a spirillum, what's peptidoglycan, what's a gram-positive cell, a gram-negative cell, what's an acid-fast stain or an acid-fast cell, what's a primary stain, what's a mordant, what's a decolorizer, what's a counter stain, what's a capsule, what's an endospore, and what's a flagella. Any question on any of that? All right, because of their extremely small size, bacteria are essentially transparent when you look at them under the microscope. Therefore, it's necessary to stain them before making a bacterial smear and then looking at them under a microscope. And how you do that is you add your bacteria to a drop of sterile water. It's placed on a slide. The bacteria is added. It's then smeared over the surface of the slide. The slide is then usually allowed to air dry. And then the organisms are fixed onto the glass by heat fixing. Any question about any of that? A simple stain simply uses one dye to stain the bacteria cell. And it says here in the lab manual, it uses a basic dye, but actually it doesn't matter if it's a basic dye or an acidic dye. The simple stain uses only one dye to stain a cell. A differential stain usually uses 2 or more dyes to provide additional information about the cells than you can obtain with the simple stain. An example of a differential stain is the Gram stain, and we'll talk about that stain today in this lab. And then lastly, there are special stains, and they're used to view particular bacterial structures, such as an endospore or a flagella. Any question about any of that? All right, this picture, can you guys see this? Can somebody confirm you're seeing this?
Yes.
Yes.
All right, this picture is showing you cocci, round spherical balls of cells, and then the different arrangements, cellular growth arrangement that the cocci can form. Cocci can be individuals, a single coccus, or they can be diplococci, 2 cocci stuck together, or tetrads, 4 cocci stuck together, or staphylococci, which that literally means a cluster of grape cocci. And that's just a cluster of cocci. And then they can be linked together in a link that we call them streptococci. So you call that a string of cocci. You can also have bacilli, which are a bacillus or a rod, and they can be single cells or singlets, single bacilli. They can be diplobacilli, and they can be staphylobacilli. They are not tetrads. And I'm trying to remember if we use the term streptobacilli. I think you can use the term streptobacilli, although most people call that a linkage of the bacilli. And we'll talk about the different links because bacilli can be linked sausage form. If you've ever bought sausage in a linkage, you know what I'm talking about, where they'll be linked together this way. And the bacilli can also be linked together this way. And we'll talk about that. They're called palisades. Besides bacilli and cocci, you can also have spirilla. And please do not call it spirilla bacilli. Nobody uses that term except for this author who made this picture. Call it a spirilla. That's a spiral or an S-type shape. That's not very good of an S, but there's one there that's much better. Okay, any question about that? Meaning this is a bacilla, this is a spirilla. And you can also have cells which are in between a cocci and a bacilli. Just have rounded bacilli and then shorten it, and we call it a coccobacilli. Okay, this is showing you another shape of a cell, pleomorphic, and the shape of the cell can take on any different shape. I'll show you a better example in the lecture of a pleomorphic cells. All right, remember a simple stain is just using one dye of any stain to color the cells. A differential stain uses usually 2 or more dyes to stain the cells. The most commonly used and probably the most important stain in microbiology is the Gram stain. You don't need to know that it was invented in 1884 by Hans Christian Gram, but you should know that it is named after somebody. And so if ever you use Gram negative or Gram positive, the G should be capitalized because it's named after Dr. Gram. He used the stain initially to differentiate between a bacterial infection and the patient's own cells. So it was differential for the bacteria from the patient's cells. But microbiologists started using it and they noticed that it's differential among bacteria cells and that we have gram-positive bacteria and gram-negative bacteria. The gram-positive cells stain or appear purple, where the gram-negative cells stain or appear pink. What's happening is the crystal violet, which is the primary dye, gets into the cells and it remains in the gram-positive cells, but it washes out of the gram-negative cells. And then the pink color comes by the second stain, which is safranin, which is pink in the gram-negative cells. So here we're seeing some bacteria stained with the gram stain, and you can see gram-negative rods here and here, gram-negative cocci here. I don't see any gram-negative spirilla. Excuse me, gram-positive spirilla. Did I say negative? That's a gram-positive cocci and a gram-positive, gram-positive bacilli, a gram-positive cocci. That one is close to a spirilla, but I'm not sure it is. And then we have gram-negative bacilli here, a gram-negative cocci there, and a gram-negative spirilla right there. And there's another one there. Gram reactions are based upon the ability of the bacterial cell to retain crystal violet. What's happening is the gram-positive cells contain a thick peptidoglycan layer, while the gram-negative bacteria, if they have a cell wall, have a much thinner peptidoglycan layer. And the crystal violet washes out of the thin peptidoglycan layer of the gram-negative cells, but it does not wash out of the thick peptidoglycan layer of the gram-positive cells. So let's look at the cells. Here's the cell wall. And as you notice, in the gram-positive cells, it's almost entirely, the cell wall is almost entirely made out of peptidoglycan, and it's very thick. You can't see the layers, but this is layer upon layer. It's hundreds of layers, maybe even thousands of layers of peptidoglycan. The gram-negative cell wall does contain peptidoglycan, but it's thinner than the gram-positive cell wall. So it's fewer layers, but it's not one. Frequently people tell me, oh, there's only one layer of peptidoglycan in a gram-negative cell. That's not correct. There are many layers of peptidoglycan in a gram-negative cell, but it's much less than a gram-positive cell wall. The gram-negative cell also has lipids on the external part of the cell wall, and we'll talk about that later. Now, why is that only the lipopolysaccharide layer? That should be including this. Oh, well, not a big one. But the point is that there are lipids that we call the outer membrane of the gram-negative cell wall. And there's a thinner peptidoglycan layer in the gram-negative cells. And this layer right here is the plasma membrane of the cell. It's not part of the cell wall. You should know that some gram-positive bacteria lose their ability to retain the crystal violet dye and are easily decolorized. Therefore, they're described as gram variable, where it will look like a mixture of gram-positive and gram-negative bacteria, like this right here. Now, I've worked with cells that weren't, well, they easily lost their crystal violet, and occasionally they would look like this. But I can tell you that if you do the gram stain correctly, even on these cells, the cell will stain gram-positive. These cells don't readily take up actually any of the dyes, and we'll talk a little bit about that later. But they do take up a little bit of the dye. And if you use the gram stain correctly, where you're using freshly grown cells, which in the gram stain, you should only use freshly grown cells, cells that are not older than 2 days old, you will not get this appearance. You will always get gram positive. And this is seen in the mycobacterium, where you see gram variable, meaning gram negative and gram positive cells. But I've only seen this when the mycobacterium are not freshly grown. So if you stain only freshly grown cells, they will appear gram positive. It's only when you use old cells that they will take on the gram variable appearance. And if you notice here, what can you say about this cell here or that cell here compared to that cell here and this cell here? So we're talking about that one right there, that one right here, compared to this one and that one. What do you notice? Come on, somebody's got to be observant.
Some are bigger than others.
Well, yes, that's not what I'm getting at. What's another thing you notice?
That they're together.
Well, some of them are together, but what's about
Different
that?
shades.
Yeah, they're different shade. That's exactly what I was getting at. This one and that one in particular are stained lighter than this one and that one. And if you look closely at this one, that one even looks broken. And that one, you'll notice they look ghostly. The cell doesn't have a specific shape, doesn't have a clearly demarcated boundary of where the cell is and where the cell isn't. And the cell looks like a ghost. You know what I'm saying? It has the outline of a cell, but the cell isn't all there. Which if you've ever seen a ghost, it has the outline of a person, but the person isn't all there. And these cells, they have the clearly demarcated boundary where the edge of the cell is. They're darkly staining. These cells are viable cells. They have a clearly demarcated beginning and end of the cell. These cells probably are dead cells or dying cells. And so the boundary of the cell isn't as clearly demarcated. They look ghostly. Does everyone see that?
Yes.
Okay, great. The mycobacterium have a lot of mycolic acid, which is actually a lipid in their cell walls. And these cells do not as easily take up the dyes of the crystal violet. And so they can take on a gram variable result. But every time I've seen the gram variable result, and I ask the student, how old are these cells? Because they don't look uniform. Some are looking gram positive, others are looking gram negative. And the student has always told me, oh, these cells, they're old. They're 2 weeks old or older. And I tell them, you can't do the gram stain on cells that are older than 2 days old. They have to be freshly grown or the gram stain may not work correctly. Any question there? And for
So how
these,
do they know when like a culture is contaminated?
well, when you do a gram stain, if that's what you're getting at, and you see this in the culture, and then these are all sharply staining pink, meaning they're not ghostly, and these are all sharply staining purple, then you know you have a contaminant. Another way would be if these were rods and these were cocci. Clearly a contaminant.
Okay.
Okay. This isn't a contaminant. The trouble is these are mycobacterium. They don't readily take up the gram stain. And then these cells are old. I can tell that because they're ghostly. And you can imagine on a dead cell, the crystal violet gets in it, but then it can easily wash out because the cell wall is not completely intact. All right. There are some tips to prevent the Gram variable. Avoid using old cells. Make sure you have a thin cell, meaning a layer of thin cells, because if you have multiple layers, cells upon layers of cells, the stain can easily get the outer cells, but the stain has a harder time getting into the cells deep down. And then when you're washing out the dye from a gram-negative cell, if you have layer upon layer of cells, the dye will easily be removed from the outermost cells, the surface cells, but the cells deep down in will be harder to get the dye to leave those cells. So make sure you work with a thin cell. Heat fix the bacteria on the slide after the smear is air dried. And then avoid overheating the cells during heat fixation. Make sure you use fresh staining reagents. And then most importantly, do not over decolorize the slide. We'll talk about that. Or under decolorize the slide. All right. And then if you do see differences in your slide, look for a region where the cells are evenly dispersed and use that to determine the Gram stain of your cells. Any question about the Gram stain? We'll talk about it more in a little bit or later in the course. Another stain that's a differential stain is the acid-fast stain. Some bacteria, such as the Mycobacterium and Nocardia, have a waxy substance called mycolic acid in their bacteria cell walls. These cells are able to resist the decolorization in the acid-fast stain. And therefore, they're called acid-fast bacteria because they stain with the acid-fast primary stain and they retain that stain, whereas the other cells, which are not acid-fast, lose, or acid-fast negative, lose the acid-fast stain. Acid-fast bacteria stain pinkish because the acid-fast stain is pink. We'll talk about what it is in just a minute. The acid-fast negative bacteria stain either greenish or bluish because of the secondary stain is bluish or greenish.
Can you describe how an acid-fast stain is different from a simple stain?
Well, for one thing, it's using two different dyes. And then it's showing you differences in the cell. And that is the acid-fast cells have a lipid in their cell wall, which does not allow the acid-fast stain to come out of the cells in the decolorization step. And then the acid-fast negative cells do not have the lipid in the cell wall. And the acid-fast stain washes out of the cells in the decolorization step. All right, any other questions about the acid-fast stain? All right, special stains are stains that we use to see something special in the cells. So some cellular characteristic or structure. A negative stain comes from the term of a negative in photography, which most of you have probably never even seen because that's the old is it Polaroid film? Is that what it's called? Something like that. Where when you looked at it, it had the opposite color or contrast of the real object. And that's why it was called the negative. And the negative stain, you stain the cell, or actually the negative stain does not stain the cell, it stains the background. And you can use a negative stain to determine whether a cell has a glycocalyx. If the glycocalyx is firmly attached to the cell wall and you can see it under the light microscope, we call it a capsule. It's a substance that is secreted by the cell. It's external to the cell wall. So we say it is outside the cell. And there's the capsule here. The green, the yellow being the cell wall. The production of a capsule can be associated with the organism's virulence or its pathogenicity, like Streptococcus pneumoniae produces a capsule, and then this makes it hard for the immune system to get through the capsule and then attack the bacteria. The capsule tends to make the colonies of bacteria take on a mucoid appearance. So these mucoid colonies are because the cells have a capsule. And then if we stain the cells with a negative stain, this is similar to a negative stain. This is actually a capsule stain. It stains both the background, the glass of the slide, and it's staining the cells. And so the background and the cell is stained dark, and then the capsule is unstained, and so it appears clear or white. And here you can see the capsule around these cells. This is Klebsiella pneumoniae, another bacteria which causes pneumonia. And well, we won't get into that. Another special stain is or can be the endospore stain. Endospores are a metabolically inactive hardy structure that's produced by some bacteria. So the bacteria can survive unfavorable environmental conditions. So when the environment starts deteriorating, some bacteria can make an endospore and then can survive when the environment becomes inhospitable to the bacteria. And then when the environment becomes favorable again, the endospore can germinate and the endospore will return to the growing bacteria, and the bacteria will return to its normal metabolically active state. And we call this the vegetative state or the vegetative cell. So the vegetative cell is metabolically active. It's engaging in metabolism. It is engaging in cellular growth and reproduction. The endospore, although it's not totally metabolically inactive, it has very little metabolism going on, and the cell is in stasis, so the cell can survive. There's only a few bacteria that can produce endospores, and most of them are in the genera of Bacillus and Clostridium. So mainly these two genera form endospores. And this table is showing you some pathogenic endospore forming gram-positive bacilli. And some of them you've probably heard of. Bacillus anthracis causing anthrax, Clostridium botulinum causing botulism, Clostridium tetani causing tetanus, Clostridium difficile causing C. diff, sorry. The endospore does not make these cells dangerous. The endospore is not toxic to us. The trouble is with the endospore, when you treat the bacterial disease with antibiotics, the endospore will survive. And so usually with something like a patient getting C. diff, the patient has to be treated twice. Once to get rid of the growing C. diff, but the endospore survives, and then the environment becomes favorable when the antibiotics is discontinued and it wears off in the patient. And then 5 or 7 days later, you retreat the patient for C. diff because the endospore will create the C. diff infection again, meaning the endospore will germinate and C. diff will start growing. And then you treat them a second time. And that usually gets rid of C. diff. All right, we'll talk about the endospore stain a little bit in the lab proper. Bacterial endospore stain green. And that comes from the primary stain, which is malachite green. We use mordant, which is simply heat usually, to get the malachite green to go in the endospore. You then decolorize the cells, just washing them with water. Malachite green dissolves in water. And so if the cell doesn't have an endospore, the green will wash out with the water wash. But if the endospore is there, let me blow that up. Oh, there it is. If the endospore is within the cell or outside of the cell, the endospore will retain the green color because the endospore keeps the malachite green from washing out. And then you stain the cells with the second stain, which is pink, and that'll stain the vegetative cells. So this is endospore positive, that cell is endospore negative. All right, another special stain is the flagella stain. It stains the cells to show flagella. Flagella are this whip-like structure in cells, which are not normally seen under a stain under the microscope. Usually the flagella stain coats the flagella many times. And so with each coating, you make the flagella eventually visible to be observed. For the lab, you need to know that the flagella can take on different arrangements. You can have monotrichous, where the flagella on one side, we call that polar as well. Lophotrichous, there are several flagella, but they're all on one side of the cell. There's amphitrochus, and that one is multiple flagella, and they're on two sides of the cell. And then there's peritrichous flagella, and the flagella are everywhere in the cells. And here we can see cell stain with a flagella stain. And you'll note that when you stain cells with the flagella stain, that they can take on a different flagella arrangement. This is clearly peritrichous, but that one looks monotrichous. And that one could be lophotrichous, meaning the flagella are all on one side. So, and that one could be slightly peritrichous, but it's mostly, well, no, that's not amphitrichous. It doesn't have it on this side. The question is, how do you read this? And that is, you have to realize that the cells put down their flagella with time. So this cell and this cell and that cell have put down all of the flagella. But this cell is only starting to put down the flagella. And this cell also only starting to put down the flagella. And that cell only just starting, it's only got one flagella. And given time, these cells will put down more and more flagella, and they probably will eventually take on this or that appearance or that appearance. So how do you read the flagella stain? You don't say it's a mixture of different trichus. You take the highest trichus, peritrichus, and then assume that the other cells, given time, will become peritrichous. Any question about any of that? All right, so know the terms of this lab. We don't need to look at the references. And then do the laboratory exercise. You're not physically going to be performing any of the activities in this exercise, meaning your worksheet, but you must understand how to prepare bacterial smears for stainings and be able to answer the following. Procedure 1, we're preparing a smear for staining. A smear is something you make before you stain the cells. And we have a video clip on making a bacterial smear. And then we have a video clip on heat fixing. So once you make the bacteria smear, usually you let it air dry. And after it's air dry, you heat fix the cells. So understand the steps for making a smear and then air drying and heat fixing. And those steps are listed in 2. Procedure 2 is looking at a simple stain. View the following clip of performing a simple stain. You do a simple stain on a bacterial smear. So you have to make the bacterial smear first and then do the simple stain. Review the steps of doing a simple stain. And then answer the questions. Let me get the worksheet up so I can show you the questions. Sorry, I've got the hiccups right now. All right, so for procedure 1, you have to answer question 3. State 4 common errors when preparing bacterial smears for a stain. And then for procedure 2, we're looking at the simple stain. And then describe the cellular morphology. That's the shape, not the cellular growth arrangement of E. coli, shown here, and Staphylococcus aureus, shown here. For B, answer what's the total magnification. And that's given somewhere. Ah, here it is. It's the oil immersion objective that's being used. For procedure 3, we're looking at the gram stain. View the following clip of making a gram stain. And then you're going to practice performing a gram stain by doing the virtual lab. Click on this link or the link in the lab module and allow it to download and load on your system, meaning it probably will take a little bit of time. And use the different colorization solution to do the step. So when making the gram stain, what you first do is flood the smear with crystal violet. Leave the Smith's crystal violet on the smear for 1 minute. Then rinse with distilled water. Second, flood the smear with iodine. Let it soak for 1 minute. And then rinse with distilled water. Third, flood the smear with alcohol acetone decolorizer, or you just say decolorize it. Let it soak for about 5 minutes and immediately rinse with distilled water. And actually, it's better if you don't do it for 5 seconds. It's best if you take your decolorizer to the slide. Can you guys see this?
Yes.
Yes.
Okay. And then you put it on the slide and you watch the decolorizer run down. Once the decolorizer, when running into the cells, loses its color, stop the decolorization and immediately rinse with distilled water. Why you need to immediately rinse with distilled water is these cells have decolorizer on them. The decolorizer will continue to work even though you've taken the, you've stopped adding the decolorizer. So you need to rinse with distilled water to stop the decolorization. And it needs to be done immediately because the decolorizer will pull out crystal violet from the gram-positive cells if you give the decolorizer enough time. Then rinse the decolorizer with distilled water, then flood the smear with safranin, let it stain for 1 to 2 minutes, and then rinse the slide with distilled water. You can then blot dry with bibulus paper. And bibulus paper is just rough paper. What you want to do, I don't have a good picture, so I'll use this as my slide. Put the slide in the bibulus paper and then blot dry. Pull out the slide and there will be so little water on it that you can add oil if you want and look at the cells under the microscope, including with the 100x lens. So that's the procedure. Let's take a look just briefly at the virtual lab. Click continue.
Hi, Lab Assistant.
Ouch.
Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
All right, click the next button. Continue the thing. Virtual lab.
Hi,
Let
Lab
it run.
Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria. Before you start, there are basic safety procedures.
Okay, any questions about that? This seems to run pretty well. Let's move on to the microscope. All right, this is going through the Gram stain. All right. Did I? Sorry, I stepped the share. Hit the wrong button. All right, let's close this. The virtual lab uses a differential decolorizing solution than we discussed. So their step is longer than we did. If you just use alcohol, which is what I'm assuming that the virtual lab is using, you use the decolorization for more than just a few seconds. If you use our decolorization agent, you decolorize only for about 5 seconds. All right, the first stain on the Gram stain, smear with crystal violet, let it soak for 1 minute. Rinse with distilled water. Second, flood the smear with iodine, let it soak for 1 minute. Rinse with distilled water. Third, flood the smear with the decolorizer. Immediately rinse with distilled water. Fourth, smear the slide with safranin, let it soak for 1 to 2 minutes and rinse with distilled water, and then blot the slide dry with bibulous paper. An easy thing to remember in my procedure is everything is for 1 minute except for the decolorization step. And then everything is followed by a water rinse. So if you're giving me the procedure, you don't need to say this, rinse with distilled water. You just, one time in the procedure, say after every step, rinse with distilled water. When you initially stain the cells, all cells stain purple. They all take up crystal violet, the Gram positive and the Gram negative. You apply Mordant. Mordant, what it is the iodine binds with the crystal violet, makes a larger molecule. It does intensify the stain a little bit, so it'll be a little darker, as well as a little bit more of a purple color, meaning a purple color, whereas crystal violet is a blue purple color. And then you decolorize the cells, and that will pull out the crystal violet iodine complex from the Gram negative cells. And so they will appear clear, but it will not pull out the crystal violet iodine complex from the Gram positive cells. So they will continue to stain purple. And then you stain with the counter stain, the safranin, and then the clear cells will show up pink. And that's the state of the Gram stain. Answer the question below are 2 smears viewed with the oil objective lens, stained with the Gram stain. So tell me, is E. coli shown on the left? Where is that statement? Right there. And Staphylococcus aureus on the right, whether it's Gram positive or Gram negative. And then tell me the cellular morphology, the shape. Are the cells coccus or bacillus? Then answer the questions, which this isn't labeled here. Where do I have that? Oh, I see why it's not labeled. Come back. That is for procedure 3. Then answer these questions. A, Staphylococcus aureus, after the primary stain is added, what color will it be? E. coli, what color will it be after the primary stain is added? Staphylococcus aureus after the acetone decolorization step, E. coli after the decolorization step. So answer those questions there. Procedure 4, we're going to look at the acid-fast bacteria stain. We have a video clip to watch on it. Review the steps of the modified Kenyon acid-fast bacteria stain. You stain the cells with carbol fuchsin, which is the primary stain, but it stains both the acid-fast stains and the acid-fast negative cells, meaning both cells are stained. And then you do the acid alcohol wash, the decolorization step, and the acid-fast negative cells are decolorized, they're clear. The acid-fast positive cells retain the carbol fuchsin. And then you stain the cells with the brilliant green, the counter stain. And the acid-fast cells will retain their carbol fuchsin, and they will stain pinkish in color. The acid-fast negative cells will lose the carbol fuchsin and pick up the counter stain, so they will appear green in this procedure, but it would be either green or blue. And then this is the different steps of the procedure. And then look at the smears. And then you need to tell me for each arrow whether the cells are acid-fast stains, meaning acid-fast positive cells, or acid-fast negative. And you can either write that in there, or you can write it, I don't know, leftmost is, top arrow is, and then for this one, left arrow is, right arrow is. But you need to demarcate it, which one is which. I'm going to answer the questions on special stains. We have a video clip going over the endospore stain and a video clip going over the negative stain, which is looking at a capsule. And then look at this slide A, and then answer the questions in A here. This is the Schaefer-Fulton stain, which uses a dye as the primary stain, but you got to name which dye it is. And ultimately it dyes or colors what, which are produced by some bacteria to aid in survival. So fill in the blanks. And the lab talks about it if you don't know. And then list some examples of pathogenic endospore forming bacteria. Meaning what's a pathogen of humans which have endospores. And then B goes to this slide here. This is a slide stain for the capsule, not the capsule, the flagella stain. And so you have to tell me what stain it is. And it coats the lip-like appendages, which aid in motility. State the flagella arrangement that you see in picture B. For B, this is what stain. It uses India ink or nickerson, which can be used to detect the presence of what in the cell, meaning the structure in the cell. And what is located exterior to the cell wall and prevents phagocytosis. So this stain is a stain that is looking for the presence of what? And then list some pathogenic microorganisms that produce a capsule. After you're done with these questions, you then have to do 2 case studies. Case study A is right here, a 64-year-old patient is admitted to the hospital with soft, excuse me, that's case 2. Let's go back to case 1. Case 1, a 45-year-old female is experiencing respiratory issues. Based upon her presentation, doctors order a chest X-ray, and they do a sputum for Acinetobacter bacterial culture. And then you do the smear, and there's the bacteria. You'll notice the lungs don't look right, correct, right there, and a little bit right there. The lung should be like that and that. And this area here doesn't look right. They didn't do a sputum smear, and there's the stain of the sputum. And what cells do we have here? And then lastly, name the species which is causing this infection. You cannot simply state what the infection is. You must name the species that is causing the infection. For example, if it was a cold, you couldn't just say human cold. You have to name the species which is causing the infection. In case study 2, a 65-year-old diabetic patient is admitted to the hospital with soft tissue necrosis right here. A routine culture and gram stain is ordered. There's the gram stain. The bacteria grow only anaerobically, meaning they're anaerobes. And based on the findings below, what's the most probable diagnosis? You should take a look at this slide here, which is using the Schaefer-Fulton stain, which is a stain that was discussed in the lab module. Look and see what is staining if you don't know what it is. And then try and figure out what species is causing this person's bacterial infection. And to do that, you need to go up to a table that was shown in the lab module. I think I've already closed it down. Oh, there it is. Yeah, it's table 4.1. So to get your bacteria, it should be one of the bacteria in this table, table 4.1. All right. So remember to state the bacteria that's causing the infection. When you're all done, submit your worksheet to the area for lab 4. Any questions? If not, I will be here until 8:20. If you don't see me, it means I stepped away from the desk for a little bit, but I will be returning, and I will be here until 8:20 to help you with the lab. So work on the lab, get the worksheet done, and then turn in the worksheet. The worksheet must be turned in by Saturday at 11:59 p.m. for full credit.